Hello students, Assalamu alaikum. Today we are going to start metabolism part 4. Let's talk about the mechanisms of enzyme action. Mostly the enzymes are the protein molecules and the catalyst for biochemical reactions means they speed up the biochemical reaction by lowering the activation energy. Specific part on the globular surface of enzyme where substrate binds and actually take part in the reaction is called the active site. The active site is usually a very small portion of the enzyme which is a charge bearing cavity having a specific shape. The action of enzymes can be explained in a few main steps. Specific substrate molecule temporarily binds to the active site of enzyme to form enzyme substrate complex. Once the ES complex has formed, enzyme catalyzes the reaction to convert the substrate into product, thus converting the ES complex to enzyme product complex. Finally, EP complex breaks up into the products and enzymes. The enzyme molecules remain unchanged at the end of the reaction and is available again if needed for the same reaction again. Enzyme action is highly regulated by factors like pH, temperature, and the presence of inhibitors or activators. Enzymes can be inhibited by molecules that block or alter the active site or activated by the molecules that enhance binding or reaction rates. The rate of reaction increases with increase in temperature but up to a limit. Different enzymes have different optimum temperature and optimum temperature for human enzyme is 36 degrees Celsius to 38 degrees Celsius. Each enzyme performs best at a specific temperature, which is its optimum temperature. As the temperature increases, enzyme activity increases because the molecules move faster, resulting in more frequent collisions between enzyme and substrate. But if the temperature becomes too high, the enzymes become denatured, which disrupt the active site and leads to loss of function. Lower temperatures slow down enzyme activity because molecules movement decreases, and at very low temperature enzyme remain inactive due to fall in available activation energy. And they will regain their catalytic activity when the normal temperatures are restored. pH of medium affect the enzyme functioning too. Each enzyme performs best at the specific pH, which is called optimum pH. Optimal pH for the human body is near neutral, that is pH 7. Slight change in the pH of the reacting medium below or above the optimum pH reduce the rate of reaction. Extreme changes in pH denature the enzyme. For example, pepsin and trypsin are both protein digestive enzymes. Pepsin perform best in acidic medium in stomach at 2 pH, whereas trypsin perform best in alkaline medium in small intestine at 8 pH. In the presence of enough enzyme molecules at the reaction site, the rate of reaction is directly proportional to the substrate available. So the rate of enzymatic reaction increases with the increase in the substrate concentration, but up to a certain limit. After that, a point is reached when further increase in substrate concentration will have no effect on the rate of enzymatic reaction. It is due to the presence of extra substrate molecules in the medium which have occupied all the active site of the available enzyme molecules. This condition is called the saturation of active site. Sometimes enzymes are not able to perform their role due to the presence and interference of some chemicals at the reaction site. This phenomena is called enzyme inhibition. And the molecules that stop the enzymatic reaction by binding with enzymes and do not form the products are called inhibitors. There are two types of inhibitors, competitive inhibitors and non-competitive inhibitors. If the enzymes are inhibited due to the presence of molecules which occupy the active site due to their similarity in structure and shape to substrate is called the competitive inhibitors, and the phenomena is called as competitive inhibition. Competitive inhibition is temporary and can be reversed if the concentration of actual substrate is increased than the concentration of inhibitor molecules. If the enzymes are inhibited by the inhibitors that do not resemble to the substrate molecules are called the non-competitive inhibitors, and the phenomena is called non-competitive inhibition. They may attach to the enzyme surface at a point other than the active site called allosteric site. They alter the globular shape of the enzyme and damage the active site of the enzyme. Thus, the actual substrate will not be able to bind to active site and reactions stop. Some molecules increase enzyme activity. They can bind to allosteric sites, stabilizing the active form of the enzyme and making the active site more available for substrate binding. These are called activators. Thanks for watching. Like the video and subscribe the channel for more videos.